SNP genotyping is the measurement of genetic variations of single nucleotide polymorphisms between members of a species. It is a form of genotyping, which is the measurement of more general genetic variation. SNPs are one of the most common types of genetic variation. A SNP is a single base pair mutation at a specific locus, usually consisting of two alleles. SNPs are found to be involved in the etiology of many human diseases and are becoming of particular interest in pharmacogenetics. Because SNPs are conserved during evolution, they have been proposed as markers for use in quantitative trait loci analysis and in association studies in place of microsatellites. The use of SNPs is being extended in the HabMap project, which aims to provide the minimal set of SNPs needed to genotype the human genome. SNPs can also provide a genetic fingerprint for use in identity testing. The increase in interest in SNPs has been reflected by the furious development of a diverse range of SNP genotyping methods. Hybridization-based methods. Several applications have been developed that interrogate SNPs by hybridizing complementary DNA probes to the SNP site. The challenge of this approach is reducing cross-hybridization between the allele-specific probes. This challenge is generally overcome by manipulating the hybridization stringency conditions. Dynamic allele-specific hybridization. Dynamic allele-specific hybridization genotyping takes advantage of the differences in the melting temperature in DNA that results from the instability of mismatched base pairs. The process can be vastly automated and encompasses a few simple principles. In the first step, a genomic segment is amplified and attached to a bead through PCR reaction with a biotinylated primer. In the second step, the amplified product is attached to a streptavidin column and washed with sodium hydroxide to remove the unbiotinylated strand. An allele-specific oligonucleotide is then added in the presence of a molecule that fluoresces when bound to double-stranded DNA. The intensity is then measured as temperature is increased until the melting temperature can be determined. SNP will result in a lower than expected trademark. Because genotyping is measuring a quantifiable change in trademark, it is capable of measuring all types of mutations, not just SNPs. Other benefits of include its ability to work with label-free probes and its simple design and performance conditions. Molecular beacons. SNP detection through molecular beacons makes use of a specifically engineered single-stranded oligonucleotide probe. The oligonucleotide is designed such that there are complementary regions at each end and a probe sequence located in between. This design allows the probe to take on a hairpin or stem loop structure in its natural isolated state. Attached to one end of the probe is a fluorophore and to the other end a fluorescence quencher. Because of the stem loop structure of the probe, the fluorophore is in close proximity to the quencher, thus preventing the molecule from emitting any fluorescence. The molecule is also engineered such that only the probe sequence is complementary to the genomic DNA that will be used in the assay. If the probe sequence of the molecular beacon encounters its target genomic DNA during the assay, it will anneal and hybridize. Because of the length of the probe sequence, the hairpin segment of the probe will be denatured in favor of forming a longer, more stable probe target hybrid. This conformational change permits the fluorophore and quencher to be free of the tight proximity due to the hairpin association, allowing the molecule to fluoresce. If, on the other hand, the probe sequence encounters a target sequence with as little as one non-complementary nucleotide, the molecular beacon will preferentially stay in its natural hairpin state and no fluorescence will be observed, as the fluorophore remains quenched. The unique design of these molecular beacons allows for a simple diagnostic assay to identify SNPs at a given location. If a molecular beacon is designed to match a wild-type allele and another to match a mutant of the allele, the two can be used to identify the genotype of an individual. If only the first probe's fluorophore wavelength is detected during the assay, then the individual is homozygous to the wild type. If only the second probe's wavelength is detected, then the individual is homozygous to the mutant allele. Finally, if both wavelengths are detected, then both molecular beacons must be hybridizing to the complements, and thus the individual must contain both alleles and be heterozygous. SNP microarrays. In high-density oligonucleotide SNP arrays, hundreds of thousands of probes are arrayed on a small chip, allowing for many SNPs to be interrogated simultaneously. Because SNP alleles only differ in one nucleotide, and because it is difficult to achieve optimal hybridization conditions for all probes on the array, the target DNA has the potential to hybridize to mismatched probes. This is addressed somewhat by using several redundant probes to interrogate each SNP. Probes are designed to have the SNP site in several different locations, as well as containing mismatches to the SNP allele. By comparing the differential amount of hybridization of the target DNA to each of these redundant probes, it is possible to determine specific homozygous and heterozygous alleles. Although oligonucleotide microarrays have a comparatively lower specificity and sensitivity, the scale of SNPs that can be interrogated is a major benefit. The Affymetrix human SNP 5.0 Genotyping performs a genome-wide assay that can genotype over 500,000 human SNPs. Enzyme-based methods. A broad range of enzymes including DNA ligase, DNA polymerase and nucleases have been employed to generate high-fidelity SNP genotyping methods. Restriction fragment length polymorphism. Restriction fragment length polymorphism is considered to be the simplest and earliest method to detect SNPs. SNP-RFLP makes use of the many different restriction endonucleases and the high affinity to unique and specific restriction sites. By performing a digestion on a genomic sample and determining fragment lengths through a gel assay, it is possible to ascertain whether or not the enzymes cut the expected restriction sites. A failure to cut the genomic sample results in an identifiably larger than expected fragment, implying that there is a mutation at the point of the restriction site, which is rendering it protection from nuclease activity. Unfortunately, the combined factors of the high complexity of most eukaryotic genomes, the requirement for specific endonucleases, the fact that the exact mutation cannot necessarily be resolved in a single experiment, and the slow nature of genesase make RFLP a poor choice for high-throughput analysis. PCR-based methods. Tetra primer amplification. Refractory mutations system PCR, or ARMS PCR, employs two pairs of primers to amplify two alleles in one PCR reaction. The primers are designed such that the two primer pairs overlap at a SNP location, but each match perfectly to only one of the possible SNPs. The basis of the invention is that unexpectedly, oligonucleotides with a mismatched 3' residue will not function as primers in the PCR under appropriate conditions. As a result, if a given allele is present in the PCR reaction, the primer pair specific to that allele will produce product, but not to the alternative allele with a different SNP. The two primer pairs are also designed such that the PCR products are of a significantly different length, allowing for easily distinguishable bands by gel electrophoresis. electrophoresis or melt temperature analysis. In examining the results, if a genomic sample is homozygous, then the PCR products that result will be from the primer that matches the SNP location and the outer opposite strand primer, as well from the two outer primers. If the genomic sample is heterozygous, then products will result from the primer of each allele and the respective outer primer counterparts as well as the outer primers. An alternative strategy is to run multiple qPCR reactions with different primer sets that target each allele separately. Well-designed primers will amplify their target SNP at a much earlier cycle than the other SNPs. This allows more than two alleles to be distinguished, although an individual qPCR reaction is required for each SNP. To achieve high enough specificity, the primer sequence may require placement of an artificial mismatch near its 3' which is an approach generally known as TAC-MMR. Flap endonuclease. Flap endonuclease is an endonuclease that catalyzes structure-specific cleavage. This cleavage is highly sensitive to mismatches and can be used to interrogate SNPs with a high degree of specificity. In the basic invader assay, a fen called cleavase is combined with two specific oligonucleotide probes that, together with the target DNA, can form a tripartite structure recognized by cleavase. The first probe, called the invader oligonucleotide, is complementary to the 3' end of the target DNA. The last base of the invader oligonucleotide is a non-matching base that overlaps the SNP nucleotide in the target DNA. The second probe is an allele-specific probe which is complementary to the 5' end of the target DNA, but also extends past the 3' side of the SNP nucleotide. The allele-specific probe will contain a base complementary to the SNP nucleotide. If the target DNA contains the desired allele, the invader and allele-specific probes will bind to the target DNA, forming the tripartite structure. This structure is recognized by cleavase, which will cleave and release the 3' end of the allele-specific probe. If the SNP nucleotide in the target DNA is not complementary allele-specific probe, the correct tripartite structure is not formed and no cleavage occurs. The invader assay is usually coupled with fluorescence resonance energy transfer system to detect the cleavage event. In this setup, a quencher molecule is attached to the 3' end and a fluorophore is attached to the 5' end of the allele-specific probe. If cleavage occurs, the fluorophore will be separated from the quencher molecule, generating a detectable signal. Only minimal cleavage occurs with mismatched probes, making the invader assay highly specific. However, in its original format, only one SNP allele could be interrogated per reaction sample, and it required a large amount of target DNA to generate a detectable signal in a reasonable time frame. Several developments have extended the original invader assay by carrying out secondary fen cleavage reactions. The serial invasive signal amplification reaction allows both SNP alleles to be interrogated in a single reaction. SISAR invader assay also requires less target DNA, improving the sensitivity of the original invader assay. The assay has also been adapted in several ways for use in a high-throughput format. In one platform, the allele-specific probes are anchored to microspheres. When cleavage by fen generates a detectable fluorescent signal, the signal is measured using flow cytometry. The sensitivity of flow cytometry eliminates the need for PCR amplification of the target DNA. These high-throughput platforms have not progressed beyond the proof of principle stage, and so far the invader system has not been used in any large-scale SNP genotyping projects. Primer extension. Primer extension is a two-step process that first involves the hybridization of a probe to the bases immediately upstream of the SNP nucleotide, followed by a mini-sequencing reaction, in which DNA polymerase extends the hybridized primer by adding a base that is complementary to the SNP nucleotide. This incorporated base is detected and determines the SNP allele. Because primer extension is based on the highly accurate DNA polymerase enzyme, the method is generally very reliable. Primer extension is able to genotype most SNPs under very similar reaction conditions, making it also highly flexible. The primer extension method is used in a number of assay formats. These formats use a wide range of detection techniques that include MALDI-TOF mass spectrometry and ELISA-like methods. Generally, there are two main approaches which use the incorporation of either fluorescently labeled deoxynucleotides or fluorescently labeled deoxynucleotides, with ddNTPs probes hybridized to the target DNA immediately upstream of SNP nucleotide, and a single ddNTP complementary to the SNP allele is added to the 3' end of the probe. Each ddNTP is labeled with a different fluorescent signal, allowing for the detection of all 4 alleles in the same reaction. With dNTPs, allele-specific probes have 3' bases which are complementary to each of the SNP alleles being interrogated. If the target DNA contains an allele complementary to the probe's 3' base, the target DNA will completely hybridize to the probe, allowing DNA polymerase to extend from the 3' end of the probe. This is detected by the incorporation of the fluorescently labeled dNTPs onto the end of the probe. If the target DNA does not contain an allele complementary to the probe's 3' base, the target DNA will produce a mismatch at the 3' end of the probe, and DNA polymerase will not be able to extend from the 3' end of the probe. The benefit of the second approach is that several labeled dNTPs may get incorporated into the growing strand, allowing for increased signal. However, DNA polymerase in some rare cases can extend from mismatched 3' probes, giving a false positive result. A different approach is used by Sequenom's Plex Snp Genotyping method, which uses a mass array mass spectrometer. Extension probes are designed in such a way that 40 different SNP assays can be amplified and analyzed in a PCR cocktail. The extension reaction uses ddNTPs as above, but the detection of the SNP allele is dependent on the actual mass of the extension product and not on a fluorescent molecule. This method is for low to medium high throughput, and is not intended for whole genome scanning. The flexibility and specificity of primer extension make it amenable to high throughput analysis. Primer extension probes can be arrayed on slides, allowing for many SNPs to be genotyped at once. Broadly referred to as arrayed primer extension, this technology has several benefits over methods based on differential hybridization of probes. Comparatively, apex methods have greater discriminating power than methods using this differential hybridization, as it is often impossible to obtain the optimal hybridization conditions for the thousands of probes on DNA microarrays. However, the same density of probes cannot be achieved in apex methods, which translates into lower output per run. Illumina Incorporated's Infinium assay is an example of a whole genome genotyping pipeline that is based on primer extension method. In the Infinium assay, over 100,000 SNPs can be genotyped. The assay uses hapten-labeled nucleotides in a primer extension reaction. The hapten label is recognized by antibodies, which in turn are coupled to a detectable signal. Apex2 is an arrayed primer extension genotyping method which is able to identify hundreds of SNPs or mutations in parallel using efficient, homogeneous multiplex PCR and 4-color single-base extension on a microarray. The multiplex PCR requires 2 oligonucleotides per SNP, mutation-generating amplicons that contain the tested base pair. The same oligonucleotides are used in the following step as immobilized single-base extension primers on a microarray. 5 nuclease. Taq DNA polymerases 5 nuclease activity is used in the Taqmin assay for SNP genotyping. The Taqmin assay is performed concurrently with a PCR reaction and the results can be read in real time as the PCR reaction proceeds. The assay requires forward and reverse PCR primers that will amplify a region that includes the SNP polymorphic site. Allele discrimination is achieved using FRET combined with 1 or 2 allele-specific probes that hybridize to the SNP polymorphic site. The probes will have a fluorophore linked to the 5' end and a quenched molecule linked to the 3' end. While the probe is intact, the quencher will remain in close proximity to the fluorophore, eliminating the fluorophore signal. During the PCR amplification step, if the allele-specific probe is perfectly complementary to the SNP allele, it will bind to the target DNA strand and then get degraded by 5 nuclease activity of the Taq polymerase as it extends the DNA from the PCR primers. The degradation of the probe results in the separation of the fluorophore from the quenched molecule, generating a detectable signal. If the allele-specific probe is not perfectly complementary, it will have lower melting temperature and not bind as efficiently. This prevents the nuclease from acting on the probe. Since the Taqmin assay is based on PCR, it is relatively simple to implement. The Taqmin assay can be multiplexed by combining the detection of up to 7 SNPs in one reaction. However, since each SNP requires a distinct probe, the Taqmin assay is limited by how close the SNPs can be situated. The scale of the assay can be drastically increased by performing many simultaneous reactions in microtiter plates. Generally, Taqmin is limited to applications that involve interrogating a small number of SNPs since optimal probes and reaction conditions must be designed for each SNP. Oligonucleotide ligation assay. DNA ligase catalyzes the ligation of the 3' end of a DNA fragment to the 5' end of a directly adjacent DNA fragment. This mechanism can be used to interrogate a SNP by hybridizing 2 probes directly over the SNP polymorphic site, whereby ligation can occur if the probes are identical to the target DNA. In the oligonucleotide ligase assay, 2 probes are designed, an allele-specific probe which hybridizes to the target DNA so that its 3' base is situated directly over the SNP nucleotide, and a second probe that hybridizes the template upstream of the SNP polymorphic site, providing a 5' end from the ligation reaction. If the allele-specific probe matches the target DNA, it will fully hybridize to the target DNA and ligation can occur. Ligation does not generally occur in the presence of a mismatched 3' base. Ligated or unligated products can be detected by gel electrophoresis, MALDI-TOF mass spectrometry, or by capillary electrophoresis for large-scale applications, with appropriate sequences and tags on the oligonucleotides. High-throughput high-throughput sequenced data can be generated from the ligated products and genotypes determined. The use of large numbers of sample indexes allows high-throughput sequence data on hundreds of SNPs in thousands of samples to be generated in a small portion of the high-throughput sequencing run. This is a massive genotyping by sequencing technology.